Okay, good morning, everyone. Welcome to our weekly grand rounds. We had a little break for the American Academy, so I hope everyone had a good meeting there. I'm excited to hear from the group this morning, led by Dr. Molly Chauchet and Dr. Rob Devenier. And I will hand the floor to Dr. Devenier to hear about what this group's been up to. It's an exciting project, and I hope you guys enjoy hearing what they've been up to. Rob, the floor is yours.
Hi, everybody. Molly Chauchet and I have been working on this project since I looked up my original email. It was March 31st, 2013. I approached Molly, and we'll hear all about Molly and her amazing team in a moment. I approached Molly because as a retinal surgeon, I felt that we didn't have the optimal solution for vitreous substitute for particularly our retinal detachment patients. And I sort of thought, what would be the perfect vitreous substitute? As all of you know, we have gases, we have oils, we have heavy oils, we have perfluorocarbon liquids, all of them with downsides, and none of them were ever created for the purposes of what we use them for. We took them from other fields and made do with them. For example, gases. Problem with gases, obviously, is that patients can't see while they're in their eyes. They have to position in awkward ways, and they can't fly in an airplane. Their lives are really put on hold for the duration of the gas being in their eye. Oils have their own problems, from emulsification to causing corneal problems, to causing pressure problems, to needing additional surgeries to take them out. And perfluorocarbons are very inflammatory and can only be used temporarily as surgical adjuncts. So I went to Molly, and we discussed what would be the perfect vitreous substitute. And to make a long story short, and we'll hear the details after many iterations, we had issues with toxicity, with inflammation, with osmotic damage. After many iterations, we really have now what we believe is the perfect vitreous substitute, and it has been patented. And we're working through various studies to get it to first-in-human studies, hopefully soon. As will also be discussed, there have been some other drugs in the pipeline, other uses in the pipeline that we've developed, and we'll hear about those too. And we will love to hear everyone's thoughts and comments and suggestions as to your thoughts, and are we going in the right direction? How could we do things better? And how do you think this will impact our clinical practices? So thanks, everyone, for being here. And with that, I'll turn it over to Molly.
Okay, well, good morning, and thank you for being here. As Rob said, I'm a professor at the University of Toronto, and have spent my career really thinking about what's the problem, what's the medical problem, and can we design a solution to fix it? And when we were given this challenge around the vitreous, we thought we could take advantage of our knowledge in hydrogels and design a new vitreous substitute. And along the way, we thought, now that we've got this amazing material, how can we build on that? And so we'll tell you stories about the vitreous substitute, which Adam Foreman will lead. And then we'll tell you about the work we're doing in the context of glaucoma, and Michael Dang is leading that. And then at the end, we'll tell you a little bit about an area in age-related macular degeneration, which John Labriola is leading. And, I want to just emphasize that we're here to listen. And so even though we're giving this presentation, we really welcome your feedback. If you're interested in working with us, please either send us an email or let Amandeep know, and he can forward that to us. And we've because we're telling you 3 stories after each one, we're going to ask, a couple of questions, and also maybe just pause for any of your questions and insights. Okay, so let's get started. We'll go to this slide, and these are the 3 areas that we're focused on. And we started a little company called Synecus, And Snakis is really incubating in the lab right now, but the goal is to enhance the quality of life through breakthrough ocular disease solutions. And as I said, there's 3 products that we're focused on. One for retinal detachment, which is our vitreous substitute. And this would be classified as a class 3 medical device. The second one is for glaucoma. This is a non-invasive, long-acting intraocular pressure reduction strategy. So really taking a well-known drug and reformulating it to last longer so people don't need eye drops. And the third one relates to dry age-related macular degeneration, where it would be an intravitreal injection of a sustained released biologic. Okay, so let's go to the next slide. And maybe Adam, I'll hand it over to you and you can talk about, I'm sure everybody in this audience knows the problem, so we'll zip through some of this stuff so that we can focus on the solution. Go ahead, Adam.
Yeah, thanks for the introduction, Molly. So thanks for being here today, everyone. My name is Adam Foreman. I'm a chemist, and I've been working with our team here for the past almost 4 years to develop our vitreous substitute technology and advance it into animal studies. So yeah, as Molly alluded to, it's really nice to be talking to an audience of ophthalmologists because you understand what the problem is. And Rob gave a really nice introduction too. So I don't think I have to spend too much time on this, but the big issue with retinal detachment surgery, if you require a vitrectomy, is that the currently used tamponades are either SF6, C3F8, sometimes silicone oil or heavy silicone oils. And they cause all these problems, the blurry vision, because the gases have a different refractive index, and then the need for face down posturing as well, or just general head posturing if you have gas in your eye. And then if you get oil or heavy silicone oil, the need to remove it at a later date, in addition to the other complications that Rob was alluding to. And when you put it all together, it's really bad for the patient. It's super uncomfortable. It disrupts their life for a chunk of time. They can't work. They can't, no leisure activities. They can't go on an airplane. It's really difficult for them. So that's the issue that we're looking to solve using more of an engineering solution. And what we've come up with is a novel hydrogel, and the hydrogel would be an alternative to gas or oil for use as a tamponade in retinal surgery. And what's great about our novel hydrogel vitreous substitute is that, first of all, it's transparent and has an equal refractive index to the native vitreous humor. So your vision wouldn't be blurry if it was administered intraocularly. Secondly, it's a biomimetic hydrogel. And what I mean by that, it has very similar physical properties to the native vitreous. And because of that, face down posturing wouldn't be required after surgery. And then finally, it's a slowly biodegradable material. And because of that, it could act like silicone oil sort of for a while because it lasts for a good amount of time, but eventually it would degrade and clear, so you don't have to come back and get a second removal surgery. And when you put all of that together, it allows the patient to have a much more comfortable recovery and to return to the normal life a lot quicker in a matter of potentially a day or two, as opposed to weeks. So what is our hydrogel SNK125? The main component is hyaluronan, which, as you might know, is a major component of the native vitreous humor. So that's how we've developed something that's biomimetic. But as you might know, hyaluronan on its own in viscoelastic materials like high visc or Helon, if you were to inject that, it wouldn't last very long. It would clear the eye pretty quickly. So you can't actually use that as a tamponade on its own. So what we've done is we've chemically cross-linked the hyaluronan using polyethylene glycol to create a stable hydrogel with hydrolytically stable cross-links. And that ensures that the hydrogel will remain like a semi-solid gel in the eye for an extended period of time and actually allow the retinal scar to heal after surgery. So this is a demo video for how our technology works. This is us using it in preclinical studies to inject into a rabbit eye. So the material comes in 2 components. It's kind of like a 2-component epoxy. And they're like a viscous liquid now, and initially we'll mix them back and forth to initiate a gelation process. At this point, we'll inject it into the eye while it's still a viscous liquid. This is a rabbit eye you could see being filled with the hydrogel. And once the eye is filled, it will gel in situ in a period of about 10 to 20 minutes. So it's very easy to administer when you consider the current standard of care for retinal surgery. It's less viscous than silicone oil, so it can actually be injected by hand or very easily by a viscous fluid pack if you prefer that method. And it's highly compatible with the current procedures for vitreoretinal surgery. So what are the properties that we've tested of SNK125 to show that it could actually be well tolerated, safe, and non-toxic in eyes? We've done a plethora of studies in rabbits and had it in rabbit eyes for up to 6 months to show that it's completely biocompatible, non-toxic, and safe to use. And we've determined through these studies that it's non-toxic to the retina, it doesn't cause any retinal damage. It's a non-swelling material, and this is super important because if it was a swelling hydrogel, it would lead to high ocular pressure and glaucoma will be developed. And this is often an issue with silicone oil and is also an issue with other hydrogels. It's a slow, I already mentioned it's biodegradable, and we've tried to determine how long it'll last in the eye in vivo and determine that it has a half-life of around 6 weeks. And finally, when the gel is present in the eye, it doesn't impede retinal function, which we've demonstrated using ERG. So digging a little deeper into some of this data, as I mentioned, we injected it into rabbit eyes. So these rabbits received a full vitrectomy and then an administration of either our SNK125 hydrogel or BSS as a control. And then at 3 different time points, 1 month, 3 months, and 6 months, we enucleated the eyes, sectioned them, and did histology staining with H&E staining just to take a look at the retinal structure. And looking at the structure of the outer nuclear layer to inner nuclear layer, you notice that the thickness remains constant over the duration of the experiment. So there's no damage causing thinning of the retinal layers. And there's also no significant difference between the treatment, the hydrogel, and the saline control. So it's completely biocompatible with the retinal tissue. We also measured intraocular pressure for the duration of the experiment, and the pressure for the average of the rabbits remained within the normal range for the duration of the experiment, and there was also no significant difference between the hydrogel eyes and the saline eyes. So this confirms our hypothesis that it's a non-swelling hydrogel and won't lead to any sort of damage associated with high intraocular pressure. Another experiment we did is we attempted to model the degradation in vivo of the hydrogel. What we did was we tagged the hydrogel with a fluorescent probe, and then we measured the fluorescence at different time points. And that's how we determined that it has an approximate half-life of 6 weeks and will be mostly, if not all, cleared between 6 to 9 months in the eye. And finally, as I mentioned earlier, we also did ERG experiments after the injection of the hydrogel, and we were able to determine through those ERG experiments that the retinal response remains proper after injection of the hydrogel for up to 6 months. Yeah, we measured the retinal activity, and there's no difference both between the retinal activity is maintained over time, and also there's no difference between the hydrogel and the saline control. So it doesn't affect retinal function at all when it's present. As I believe Rob mentioned earlier, our hydrogel is a novel composition of matter, and we have filed a patent on it. So that is very exciting for us. And I think this is the end of my portion of the talk.
Yeah.
So Molly, you want to take over from here?
Yeah, thank you. Okay, so we have one question that we wanted to ask. Amandeep, could you put that up, please? So you can see the first question is, would you recommend a tamponade that didn't require head posturing and or removal surgery? So just yes or no.
Yeah, I'll let that run for a couple seconds, but I think this should be the most lopsided poll in the history of our grand rounds, right?
Well, Maybe they wouldn't. And it looks like there was, okay, awesome. Thanks everybody for voting.
Well, I wanted to say my original idea was that it would be something that could potentially stay in the eye forever. And we realized there's nothing that's safe that could stay in forever that wouldn't cause issues. However, by having something that stays several weeks, as we described, it virtually replaces every situation with gases and oils, with the very rare exception of when we need to leave silicone oil in permanently. And that's, in my estimation, way less than 1% of retinal detachment. So the vast majority of cases that would otherwise need oils or gases would be replaced with this. So.
Okay, and we've got a couple of questions in the chat. Thank you. So Alejandro Oliver asked, can the hydrogel treat inferior pathology? Is it injected under air? And does it reduce cataract formation? So Rob, could you?
So, hi, Alejandro. Alejandro is one of our former fellows who's in Timmins. So nice to hear from you. The answer is yeah, because what that epoxy thing that you saw being mixed together, when it mixes for about 5 minutes, it's liquid and it's therefore easy to inject. But then it forms this semi-solid gel. It literally is like if you had it sitting on a table by itself, it would maintain its form. So yes, it does tamponade inferior pathology. In terms of doing an air fluid exchange, yes, exactly. So the learning curve is 0 for the surgeons because we do the surgery exactly the same way. If there's a detachment, we do a vitrectomy, we remove the vitreous, remove the traction. We do an air fluid exchange, which as retinal surgeons we love because a retinal surgeon gives an amazing wide field view of the peripheral retina, and we really wouldn't want to lose that. And then the laser is applied the same way, and the only difference is at the very end we inject this gel. Was
Okay,
that all the questions? I think so.
No, there's two more questions. So James Osterreicher, I hope I pronounced your name properly. Thank you for your question. Is there an art filling the eye without overfilling? I don't know if Adam, you've watched a number of our vitreoretinal surgeons do this procedure. Did you want to answer that?
Sure, and maybe Rob can also chime in if
Yeah.
he doesn't like what I say. But
Yeah, I usually like what you say.
yeah, I mean, I think that if you do overfill it, the gel will just kind of pop out of the exit ports. I've actually seen that in the video before. In my experience with retinal surgeons doing this on animals, they usually have a pretty good sense of watching the gel fill the eye and can get it almost full without overfilling it pretty well. But I have seen a scenario where the excess gel just exits the exit port while it's being injected. Rob, would you agree with that?
I mean, there's an art, but it's exactly the same. When I say there's no learning curve, there really isn't. It's injected exactly the same way as we would inject silicone oil, for example. In an air-filled eye, it drops down, as does silicone oil, and then we always have a vent in one of the ports. So we see when the oil or the substitute is reaching filling, and we can see it start coming out the port, which means it's reaching the posterior surface of the iris. And if the pressure is too high, we take a little bit out. Retinal surgeons, we pride ourselves on being extremely accurate at measuring intraocular pressure with our fingers. And we always, at the end of the surgery, make a judgment and make sure that the pressure is in a reasonable range, which is usually in the 10 to 15 range. We're careful not to overfill. If we did, for some reason, have a problem with pressure post-op, some could be removed, but that should not be the problem because, as we heard, and we've been very careful about the fact that it does not swell.
And actually, thanks for that answer, Rob. And actually, one thing we also tried, sort of on the other side of the equation, one time we were doing the surgery, we intentionally left it slightly underfilled, and then after removing all of the ports, had the surgeon take a 27 gauge needle and do a tiny top up with the gel just to fill it up, like might be done with gas, for example, and that worked really well. So there is quite a bit of tunability with our ability to handle this hydrogel.
Yeah, and I do want to just acknowledge that we've had the privilege to work with a number of very skilled vitreoretinal surgeons in the Toronto area. So we're grateful to all of them. And especially Dr. Peng Yan, who really started us out on this journey.
Thank you, Peng.
There actually are 4 more questions, and so let's see if we can get through them because I love the engagement. Thank you. So Peter Curtis says, congratulations on a great body of work. I've been eagerly waiting for this to be available. The nice thing about gases, silicone, or densron is that they float or sink and push the retina while they are there. Does this push on the retina or does it just fill the space in a more solid way? I guess there's a quick answer. Adam, do you want to take that?
I would say the latter. I would say it fills the space in a more solid way because it's a water-based hydrogel. It wouldn't, push on the retina in the same manner as a hydrophobic substance, which has an interfacial tension, but it does fill the space very well and seal the tear in that manner.
Yeah, and we did design it to have a density heavier than water, so it's not floating there. So I do think there will be some pushing on the. Rob, did you want to add, or should we go to the next?
Yeah, no, I agree. It's a great question, Peter, and I think you guys answered it well.
Okay. Myrna, thank you. Myrna Lichter asks, does it have any impact on intraocular pressure? And I think that was the data that we showed that, and the good news is that it doesn't. Yeah, so you can see here that it's within the normal range throughout, this is a 6-month period, so half a year, which is how long the study ran for in the rabbit eye. So
yeah, the good thing about the hydrogel is it degrades really slowly, and as it degrades, it doesn't swell. So it's not like it's adding more water to the gel and causing the pressure to go up. It's just going to degrade naturally over time and be replaced by aqueous humor.
I just want to add, Myrna, I'm always amused when I think back a couple years ago, Molly and I were at the American Academy, and the night before, one of the other groups, I believe from Australia, was going to present their first in human data on their vitreous substitute. And when Molly looked at the composition of it, even before the presentation, she said, that's not going to work. That's going to swell and they're going to have problems with intraocular pressure. And sure enough, and quite amusingly, when this group presented their first in human data the next day, that was the big problem, that these patients had pressure problems. And so the chemistry is obviously key.
yeah, thank you. Okay, so 2 more questions and then we'll move on to the glaucoma story. Are there any concerns with retinal displacement using this substitute? Yeah,
I would say it's unlikely because even when it's being ejected, it's a very viscous fluid. So it's got enough viscosity that it's not going to get underneath that retinal tear. It's not going to pass through, and it becomes a solid so quickly that that's not something that we've seen in any of our studies, and don't believe it's going to be a concern. Rob, would you agree?
I agree with that. And Miguel, the beauty of this substance is we don't need it to do much. All we need to do is stay there and not cause trouble, because we put it in after a vitrectomy, after the retina's been reattached, after an air fluid exchange, after all the lasers. So the retina's in position. We don't need it to do anything to the retina other than to hold it there and to allow for the laser to create a good retinopexy. And then it goes away. But we're not expecting or planning for it to in any way manipulate the position of the retina.
Yeah. Okay. Thank you. Yeah, we did think of these are great questions, and they're all things that we designed for or against to make sure that we didn't have these problems. So it was really nice. Yeah. Gosh, I can't remember. Didn't remember it was so long ago, Rob, that we first chatted, but it was really good to have a clear set of design criteria so we could design for them. The last question is from Aman Longshu. Can it be used in the office? Rob?
I mean, it could be used in the office if we could do a vitrectomy in the office, which I've certainly never had a reason to try and explore, not in the office, but in a treatment room, I guess, in an office setting. So if someone has an OR in an outpatient facility, like places in the US do, and some places, I guess, in Toronto do, yeah, it has to be done in an OR because it has to be done post-vitrectomy, post-air fluid exchange, post the retina being attached. So it can be done in an OR wherever that is.
Okay, well, thank you all. We really appreciate these questions, and I hope you will have as many questions for our next presentation, which is around glaucoma. And Michael, I'm going to hand it over to you. Thank you.
Thanks, Molly. So hi, everyone. So my name is Michael Dang. I'm a PhD candidate at the University of Toronto, where my research really focuses on developing this long-acting drug delivery system for delivering anti-glaucoma drugs. So I'm very excited to present what we've been working on, building up the technology of what Adam presented. So as you may know, of course, glaucoma affects over 80 million people worldwide right now, with current treatments relying heavily on daily eye drops to reduce the intraocular pressure. However, this approach has a significant limitation, such as more than 50% of the patients struggle to adhere to the daily regimen, but also that the fact that 1 out of 3 patients cannot properly self-administer the drops itself, leading to really, in the general field, the suboptimal treatment outcomes. So that's kind of what prompted us to see this urgent need to provide a more effective, but also more patient-friendly therapeutic options to better manage glaucoma and prevent vision loss. So the current sustained release implants for glaucoma that would overcome the aforementioned limitation are largely right now still ineffective due to several key limitations. First of all, they often require monthly invasive surgeries, which increase the risk of ocular infections. On the top of that, many of the materials that is used currently in these implants are either incompatible with the eye, being either permanent or releasing harmful acidic byproducts, such as the use of PLGA as a polymer. And on the top of that, these implants offer also limited therapeutic options. So if you notice in the field of sustained release implants, typically they only provide the sustained release of prostaglandin analogs, which restricts really the effectiveness in comprehensive glaucoma treatment. So to overcome this limitation, we developed the second product that builds on to what Adam presented. So we call it SNK225. It's a periocular long-acting hydrogel depot this time for the sustain of timolol, which is a commonly used beta blocker to treat glaucoma. So this technology is delivered through a simple subconjunctival injections, which eliminates the need for surgery in this case. The hydrogel is biocompatible and bioresorbable, as you've seen in previous studies. So it doesn't need to require any removal or release of toxic byproducts in this case. And also SNK225 provides the sustained release this time of beta blockers, as I mentioned, which this time will provide a novel class of sustained therapeutics that address the limitation of the current sustained glaucoma treatment. So to give you an understanding of how does that work, so we conducted a 2-month efficacy study in normotensive long evidence rats. The reason for normotensive is really because of timolol has previously demonstrated to also have an IOP lowering effect in animal. So in this case, I wanted to kind of give you an understanding. So on the left, you can see really a standard bolus injection of timolol malate, which you can see that upon injection, the timolol malate quickly coming out of the space after a couple of seconds. And you can see that on the right, when we inject the hydrogel within that space, the hydrogel itself really allows it to maintain the drug, which really maximizes the local drug bioavailability, as you don't see any apparent viscous or jelly substance coming out of that space. So we shown that SNK 225 has been shown to really be biocompatible and safe. So this is demonstrated using H&E staining, showing corneal and corneal epithelium thicknesses across all treatments, including saline, hydrogel only group, and also timolate and SNK 225 itself. We also looked at the PK study where we've shown that SNK225 were able to significantly reduce the systemic drug exposure when we sample blood over the course of 2 months in rat tail. So here I'm just only showing the 6-hour time point where you really see that the timol blood levels when measured between timol malate group and SNK225. And you can see that the data shows that SNK225 is really able to lower the systemic exposure to timolol compared to a bolus injection of timolmalate, where it has been previously shown to be cleared very quickly into the blood system. So in terms of the efficacy, so we monitored the IOP over a period of 2 months. And demonstrated that it has a long-lasting efficacy for up to 2 months with a single dose, where you can see that with an injection of the SNK225, we're able to lower the intraocular pressure. As you can see first on the left graph, a bolus injection of the timolmalate only reduced the IOP, but only up to 6 hours. You can see that baseline, that IOP coming back to baseline after 24 hours. And in contrast, with the hydrogel, you're able to maintain a consistent IOP lowering effect up to 49 days, as you can see on the right graph when we normalize the IOP to the day 0. That really show the extended duration being maintained and the potential for SNK225 to provide a better options in terms of sustained glaucoma management treatment. So yeah, again, happy to also answer any questions and to start any discussion that would be helping us advancing this technology.
Okay, thank you. Michael and Amandeep, we have, I think, 4 questions now that we wanted to, maybe the polls won't be so lopsided, but maybe they will.
Sure, here you go.
Okay, so the poll question number 2 is, which medication or class of medications do you prescribe as a first-line therapy? So if you could just choose one of these.
I'm just gonna let that run for a couple more seconds there.
Okay, thank you.
We are getting prostaglandin analogs as
Yeah, well, that's what we would have predicted, so that makes a lot of sense. Okay, maybe we can go to the next question. I mean, that's supported by the literature. Okay, what do you think are the biggest issues with the current standard of care for glaucoma? So in this case, you can check all that apply.
all right, so I'll end that there for you.
Okay.
So get the top 2 there for getting their drops. So I gotta say that's a big one, and irritation is something we hear a lot about.
Okay, that's really great to hear. Okay, yeah, consistent with what we've heard as well. Okay, good, thank you. Let's go to the next question. Okay, this is a simple yes/no answer. Would you consider using a sustained release system that is injected into the periocular space under the conjunctiva? I mean, really, there's 2 questions here, and maybe we can get to it in the discussion. But this one is really trying to understand whether you would go on to, inject in the subconjunctival space.
Pretty lopsided here as well. So there's a lot of support.
Oh, really? Okay, good. That's great. That's good to know. We didn't predict this one, so it's nice to learn. Okay, let's go to the next one. Thanks. Oh, I think Yeah, I think there's one more, right?
Yes.
Glaucoma one.
Oh, um,
Is there?
yes, hang on. Here you go.
Oh, okay. Okay, yeah. And so this is our last question. If there were a subconjunctival long-acting implant available, what do you think the minimum release time should be for it to be useful? So,
As that runs, I was going to ask you guys a question sort of related to this. I think
yeah.
The data Michael showed was, it was, what was it, 49 days?
Yeah,
Just under
7 weeks.
How long would, like, given the data that you have, how long would you anticipate, or what would be the frequency of these injections, based on what you see now with your model?
Yeah, with the model right now, it's a normotensive model, so it's hard to really see the efficacy itself of like how well it would be able to to lower the IOP in the actual disease state. So right now, so far, looking at this data, I would say every 6 weeks. However, because we're formulating timolol, so we're able to modulate the release based on really the linker that we're providing. So that's why we love to hear from the audience to understand what's the need out there, so that we can really cater our reformulation into the release kinetics for that.
Well, you see a mix here,
Yeah, that's
the
interesting.
minimum release time.
Yeah. I mean, I guess maybe a follow-up question, and maybe this can come in with the discussion, is our impression is that patients with glaucoma are seen every 6 months. And so that's why we were wondering, do we need to formulate this to last longer? Or because I think at least at this stage, and again, as Michael said, this is just a simple model and we have to do more preclinical studies. But if it was lasting 2 months, that would mean you would be seeing your patients every 2 months. And that was something we had heard, maybe it should last longer. But okay, this is very helpful.
I just want to say, I think it's sort of an interesting question and a difficult question to answer based on how it's posed, only because the answer depends on how many drugs the patient's on. If the patient needs to be on 3 drugs, 3 drops or 4 drops, having one that you can inject subconjunctivally and not need to do daily doesn't help that much. But if the patient can be controlled with just the subconjunctival injection, even for a month or 2 months, that would be looked upon very differently if they don't need to use any other drops.
Mm-hmm.
Yeah, I was gonna say exactly what Rob said. I think, those patients who are seen every 6 months are usually the ones who have mild glaucoma. The patients with advanced glaucoma may be seen every 3 or 4 months, and they're usually on multiple classes. And so, if you had a drug delivery system that could deliver multiple classes, then that would be great. But for a single drug, you're looking at patients who have mild, maybe moderate glaucoma, and they are seen every 6 months, but they may be willing to come in more frequently. We do have patients coming in regularly for intravitreal injections, for retinal disease, and so I think there is precedent in our practices for something like that. Specific for glaucoma, I do have a couple questions, and this may be early in your work, but something for you guys to think about. I'm not a glaucoma surgeon, but we do have Dr. Burt here. But those guys are always worried about the conjunctiva and the health of the conjunctiva, and the need for good conjunctiva for future filtering surgery, for example. And so something to consider, and I don't know if you've looked at, is what is the impact of this injection on the overlying conjunctiva and the health of that conjunctiva? And then the other way, did you notice any scleral thinning or necrosis? Because sometimes you see that with glaucoma drainage devices. So just a couple comments / questions for you guys.
Yeah, that's a very interesting question. Sorry, Molly. So yeah, that's a very interesting question to ask, and it's something we would definitely consider in the future, especially with the re-injection of the technology over time. Does that cause scarring tissue or fibrosis? That's definitely something to look at. But yeah, thank you for that very interesting question.
And then Dr. Burt actually is here, and she asked, is there any thought to creating a multi-class injection, multi-drug injection?
Yeah, that's a great question as well, and that will be definitely the next plan. We do see a lot of combination therapy, of course, in the clinic. So that's something we are planning on in terms of trying to pack 2 drugs or even more within the same hydrogel. So that's kind of the versatility of having kind of a scaffold hydrogel so that we can pack different drugs in there. So that's something we're going to test in the future in order to answer that question.
Timolol was just the first one that we tried. It's a long tested drug. It's off patent, but yeah, the plan would be to potentially have all kinds of medications delivered like this as a single bolus.
Yeah, and I guess one thing that we didn't really emphasize here is that we've formulated timolol in a unique way, where we're just getting ready to file a patent around that as well. So there is some patents that have already been submitted and another that we will submit to protect around the novel formulation. Okay, any other, thank you, Catherine Burt, for your question. Are there any other questions from the audience? And of course, Amandeep, thank you for your question. Okay, Eugene Liu wrote, it would be great for post-op cataracts. Rob, did you want to comment on that?
Yeah, absolutely. Because it would be very easy just to give a little injection, because sometimes post cataract surgery or vitrectomy surgery, also the pressures can spike for various reasons in the short term because of the viscoelastics that are put into the anterior chamber. So to give a subconj injection quickly, just like we give subconj antibiotics, would we think guard against potential post-op spikes.
Yeah, and just to build on that, there's potential for dropless cataract surgery with something like this. If you had, delivery, and I think that's what Eugene's getting at, is if you could deliver the antibiotic, the NSAID, and the steroid for sustained delivery, we may get patients off drops entirely following cataract surgery. And
right, and that's exactly what Eugene just said.
Yeah, perfect. Okay, well, I think, and another actually MD PhD student in our lab has been Leah Huo has been looking at doing some work with Peng actually on NSAID delivery, but not quite the same thing. Okay, well, given the time, why don't we move to the next discussion? So this is the shortest of the 3, but we'll hand the baton to John Lobriola, and we have just a couple of questions after his presentation. So go ahead, John.
Thanks, Molly, and thanks everyone for being here this morning. As Molly mentioned, this is going to be a little short. It's very early days, and we're kind of looking at AMD, which I don't think I need to go on too much about how it's a very complex disease, and there seem to be several gaps in our research and our understanding from our perspective. But I think one of the things we definitely want to achieve today is understand from your perspective where the critical gaps really are and how we can fill them with our technology. So I won't, again, I'll go through this slide a little quickly because I think for this audience, we don't need to spend too much time, but AMD is a complex progressive disease that has several phases based upon the issues that the patient are facing and where the problems are. So when you have the healthy retinal structure, you have the light sensing neural tissue on the innermost part of the eye, and then you have the retinal pigment epithelium that separate that from the choriocapillaris, the vasculature. And so dry AMD really starts between the RPE and the vasculature and Brooks membrane, where you start getting oxidative stress and inflammation and deposits of drusen there. And that will eventually cause the RPE to begin to atrophy, as well as the light sensing photoreceptors. And I think at this point, the patient will start to lose perhaps night vision. Eventually, as the lesions grow larger in the photoreceptors, you'll start to see geographic atrophy. And this stage is generally colloquially known as dry AMD. And then as that progresses, eventually the RPE breaks down completely. You begin to get neovascularization and growth of the blood vessels into the photoreceptor layer. And that's when you move on to wet AMD, and the problems with vision are really exacerbated, such as loss of central vision and visual acuity. So from our perspective, there are a few issues that can be addressed here. Of course, the wet AMD treatments out there are very well known, such as Eylea. So you can get these monthly injections to treat the neovascularization through anti-VEGF action. The issue with these is there's rapid clearance of the therapeutic. It's a biologic that diffuses quickly. There are other potential solutions available, but again, because these are protein biologics, they're not very stable. And so all of these issues lead to frequent administration of these drugs. And then in terms of early stage AMD, dry AMD, there aren't really any options available except up until recently with the approval of Syvor. And again, the issue there potentially is that it has a single mechanism of action, which is that it treats the immune response and not necessarily the neural tissue themselves. So what we've done is we've taken this hydrogel and we formulated it in such a way that it can stabilize a growth factor called IGF-1 within the gel. And it does it by sticking to it, by interacting with it directly. And this allows for several things. It can allow for extended release, as we described with the glaucoma project, although the mechanism is slightly different. It stabilizes the biologics so you don't have issues with degradation, and this improves the bioactivity. And we chose that factor, IGF-1, because it suggests that it has both anti-inflammatory and neuroprotective effects. So it can act on both the immune cells, which are activated in these diseases, as well as the neural tissue to help prevent them from atrophying. So as I mentioned, it is early days, but we've done a couple experiments to demonstrate that this could be an effective strategy. The first one, as you can see on the left, is we took IGF-1 just formulated in saline, so bolus injection, we delivered it intravitreally versus this product, which we're calling SNK-325. And this was done in a healthy mouse. And then a day later, we looked at whether IGF-1 is still activating its target receptor. And so you can see with the saline injection, you get rapid diffusion. It leaves very quickly, and we don't see any activation of the receptor. Whereas with the SNK-325, we still see robust activation of the receptor even after a day. And then in the experiment on the right, this isn't a head-to-head with the control of a bolus injection, but we took a mouse that has a mutation that causes retinal degeneration, progressive retinal degeneration over time. It actually mimics retinitis pigmentosa in this case. It's difficult to find a good model of AMD for mice. But nonetheless, if you just treat with control saline versus this SNK-325, and then single injections, and then measure retinal function by ERG over the course of several weeks, you can see that it looks like SNK-325 is preserving that retinal function. And so, again, this is quite early days, but it looks like with this improved activity, we can sustain the preservation of the retinal tissue over time, and we want to sort of extend these experiments to understand how long that can last and really test it against a bolus injection of a therapeutic. And so I think that kind of wraps it up. Again, it's very early days, but we do have some pulley questions to understand, whether we're going in the right direction and what, from your perspective, we can really focus on to make this something useful.
Okay, thanks, John. So, Amandeep, we have two questions. So at what point do you typically diagnose AMD? If you could just choose one of these 5 options.
I mean, we've got a group of ophthalmologists here, so we've got a very lopsided answer once again, so I'm going to cut this one short.
Okay. Okay. So, okay, interesting. That's good to know. Thank you. And the second question is, which delivery method would you prefer for biologics in AMD? So we know that everybody does intravitreal injection right now, so we have, but maybe you like that. So it'd be good to know, like, if you could choose one of these, which one would you choose versus which one do you use?
I was curious to see the results of this question, and the early results are pretty lopsided. So again, I am going to end it here and share the results with you. Eye drops seem to be the most preferred solution still. And I think it speaks to the minimally invasive nature of it. Of course, we are familiar with intravitreal injections, and they work very well. But interesting to see the results here.
Yeah, it is. I mean, I guess if eye drops could work, then why wouldn't you use them? I guess even with the compliance issues that we've seen with glaucoma, they are just so much less invasive. Okay.
But again, that's a nuanced question because it would depend on how long would the intravitreal efficacy last too. If it lasts 6 months or a year, that might be preferable to most people to daily eye drops too.
And I mean, we're assuming there's a lot of assumptions in terms of length of delivery, but also how well it's tolerated, right? Intravitreal injection every 6 months is likely fairly well tolerated. Eye drops with lots of preservatives, for example, and years of use that we see from the glaucoma patients are not always so well tolerated. So I imagine that there is some space for these other options, especially if they can be timed relatively infrequently and are otherwise
and also how they're tolerated. The reason pegcetacoplan is such a disaster in the US is not only that it needs monthly injection, but there's unacceptable side effects that are emerging too. That's the new drug for geographic atrophy. So it really depends on a lot of things.
Okay. Well, there's a comment from Miguel Cruz Pimentel. So thank you, Miguel, for your comment. We appreciate your enthusiasm, and certainly one of the things I said at the beginning, if you or others in this audience are interested in working with us, or just engaging even in terms of the clinical translation or any of that part, then please let us know. I don't know if there's any other comments or questions.
There was a question earlier. Alejandro had a three-part question. I think we missed the third part. And this is going back to the first case of the vitreous substitute. Often there's a risk of cataract. And Rob, do you know, does the vitreous substitute reduce the rate of cataract formation, or would it be similar to a vitrectomy anyways?
To me, I realized too, I forgot to answer that, Alejandro. We haven't seen that it increases cataracts. I don't see any reason why it should decrease it. Cataracts, as we all know, are funny things. They develop just when you do a vitrectomy. They develop even more so when you do an air fluid exchange. They develop more when you put a long-acting gas in or an oil in. To me, it's really a non-issue because in virtually all of these patients, assuming that they're post-presbyopic, which most of them are, I just do a phaco at the same time. So my plan will not be to put this into many phakic patients, only because I do a better job doing vitrectomy and repairing a retinal detachment when they're pseudophakic, and they're going to need a cataract operation anyway. That being said, no, we have not seen it have any deleterious effects on the lens.
Yeah, the only thing is the rabbits have a very big lens. So we have to be careful not to nick it. But But in the human, I don't think that would be an issue.
And recently we did do some surgeries in mini pigs, so which have eyes very much like humans.
Yeah. Okay. Any other comments or questions from the audience? Or anything from our team that maybe you wanted to say that you forgot to say? Okay.
Well, Molly, I do want to thank you guys. Molly, Rob, I think you guys have a fantastic team. That you, Adam, Jonathan, and Michael are clearly very talented individuals. So you really do have an all-star team here. And I echo what Miguel said. I think one of the things that draws many of us to ophthalmology in the first place when we're medical students is the degree of innovation that takes place in our specialty. And we've seen some of that today. So I do thank you guys for sharing your work. I'm excited to see where this goes. It's going to impact the cataract space, the glaucoma space, the retina space. So congratulations on all of your work, and I look forward to having you guys back in a couple of years with an update.
Perfect. Thank you so much.
Hopefully we'll be doing it in humans by then.
Yeah, yeah. Thanks everybody for your engagement as well. Thanks, Amandeep, for your hosting.
Thank you, everybody.
Thank
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 you.
you, everyone.
We'll
Bye.
see you guys next
Bye.
week.